Hi everyone, thank you for being here again. I'd like to introduce, Our research project is called Nuggets of Hope, using coral microfragments to determine best practice reef building techniques. I'd like to introduce our research team. My name is Caitlin Cambridge.
Sophie Capello.
Jack Kenny.
Henry Clark.
Haya Ross.
Coral reefs are biodiversity gold mines. They are coastal marine ecosystems that cover less than 1% of the seafloor, but are necessary habitats for over 25% of all marine species. Corals are really slow-growing animals. They provide many ecosystem services, such as reducing wave height and energy during natural disasters and storms, building up the land by producing sand, and also support tourism and recreation, the country's top industry. They're extremely important, but unfortunately they're degrading. Many factors aid in the degradation of coral reefs, including but not limited to climate change, pollution, legal but unsustainable fishing, and the introduction of invasive species such as the lionfish. Coral restoration is a process of assisting in the recovery of coral reefs. In coral restoration, specifically in the Bahamas, the most commonly used coral restoration technique is the use of coral nursery trees. These trees have been used for over a decade and are commonly used with branching corals such as staghorn shown here. In our research project, we've decided to use a new approach that has never been done before in the Bahamas called microfragmentation. Our main species focus will be on mounding corals, as these corals are key reef builders. They make up the bulk of the Caribbean reefs, and they also help to create a base layer for all other corals to grow.
So what is microfragmentation? It's a process of collecting 10% of a coralette from a reef, taking it back to our lab, and cutting it into 1 centimeter by 1 centimeter pieces. As we know, corals are very slow-growing animals. One fin kick in the reef can destroy 50 years of growth very easily. So we do microfragmentation because when we cut these corals into smaller fragments, they believe that they're young again. And just like human kids, they grow at an exponentially faster rate than they do in adulthood. This means that we can outplant our corals faster to the reef, which encourages biodiversity and just benefits the overall health of the ecosystem. Currently in the Bahamas, we have a lot of declining reefs. As you can see here, there's lots of red and yellow. This means that our reefs are impaired and in poor condition. So typically, a coral reef, to be healthy, should have over 30% coral coverage, but unfortunately we have less than 15%, which just goes to show that our reefs are declining. Based on the poor quality of our reefs, we wanted to assess which setting the coral nursery, wet lab, or open ocean reef best promotes the wound healing, survival, and overall health of our 3 coral species in our coral restoration project.
So the 3 coral species that we are going to be using for our project are first staghorn coral, or the scientific name Acropora cervicornis, next mountainous star coral, or Orbicella fabiolata, and finally massive starlet coral, or Psittacea psittacea. And so the reason we chose these 3 corals was because they are endangered in the Bahamas, and additionally they are key Caribbean reef builders. So first what we did is we would go onto the reef, as you can see here, and we would harvest the corals. Next we would go back to our lab, and using a diamond saw we would cut the pieces of coral into microfragments. And with those microfragments we would place them onto our hard limestone substrates, or as we call them, cookies. And so within these 3 coral species we tried to determine which treatment setting promoted the most healing and growth for our corals. And so the 3 treatment settings are first the wet lab here at CEI, as many of you have probably seen. Next, the nursery, suspended nursery in the ocean. And finally, third, the reef itself. So initially we thought the wet lab would promote the most healing and growth for our corals because its closed environment allowed us to monitor the corals more closely and filter the water. In turn, we thought that the nursery tree would fare better than the reef because its distance from predator fish such as parrotfish, damselfish, and butterfly fish would help it survive. So over the course of 6 weeks, we took our data collection on 3 factors. Each week we would determine the survival of the corals, and that's, did the corals survive, yes or no, and are there any missing microfragments? Next, we would look at wound healing. So as you can see here, where the white face of the coral is showing, that's where we actually cut it with our diamond saw. And we would look for polyp overgrowth. So polyps, as you can see here, are the actual coral animals themselves. So we would try to gauge a percentage of growth. And finally, we would look at algae overgrowth. So algae is detrimental to corals because, particularly at young stages, it suffocates them from growing. So what we would do is we would take basically steel toothbrushes and we would clean the algae, and this would help with our survival.
So in terms of our results, as you can see from this table, we have the survival of our 3 species in terms of a percentage. So 100% of our microfragments survived in the wet lab and nursery treatment for all 3 species, which is pretty impressive. Our reef treatment was not quite as successful. A few of the fragments underwent mortality, but the majority still survived. So this graph shows our wound healing for our staghorn coral species. On the y-axis is the wound healing in terms of percentage. That's the average for the treatment setting. The time in weeks since outplanting, since we cut the corals and put them in their treatment on the x-axis. And the colors of the lines correspond with the legend, but in case you can't see, the green line is our wet lab treatment, the yellow line is our reef treatment, and the red line is our nursery treatment. You can tell from weeks 2 to 4 that the nursery treatment exhibited the fastest growth and reached a wound healing average of 100% by week 4. Our wet lab was also quite successful, but it took till week 5 to reach that 100% average, so slightly slower growth rates than the nursery treatment. And you're probably wondering why there's downward wound healing. A lot of our fragments on the reef underwent predation, as Jack talked about, and some of them were swept away by the current. So that was very detrimental to the average and actually brought it down. This is a similar graph, except this is for our mountainous star corals. Similarly, we have the wound healing on the y-axis, the time in weeks on the x-axis, and all the line colors are the same. Again, the nursery treatment had the fastest growth, reaching about 80%, but not quite 100 for this species. The wet lab treatment was also successful, but again, slower growth rates. And the reef treatment, there was a sharp downward spike from weeks 4 to 5, and no growth from weeks 5 to 6. The massive starlet coral is different than the other two because the wet lab treatment actually worked the best. It showed the fastest growth, the most steady growth, no negative drops or any spikes in the data, and the highest final average for wound healing. The other 2 treatments weren't very successful. Both of them had some pretty large spikes from weeks 3 to 4 for the nursery and 4 to 5 for the reef. But we hypothesized that this is due to algae overgrowth. So our massive starlet fragments experienced a lot of problems with algae overgrowth, and having them in that wet lab right here at CEI allows us to clean them very easily, so we could give them that extra aid to make sure that they're growing in the optimal treatment setting.
So building off of what Henry just said, algae is a very important health indicator of our microfragments. As Jack mentioned before, algae suffocates a coral and actually prevents it from growing. So the graph you see before you represents algae overgrowth in our 3 species. The x-axis is time by weeks, and we had 6 weeks worth of data, and the y-axis is the percentage of algae overgrowth. In case you can't see, the green line is staghorn coral, the red line is mountainous star coral, and the white line is massive starlet coral. So some of our findings include that there is very little algae overgrowth in staghorn coral and mountainous star coral overall. After week 3, there was an incline in algae overgrowth in all 3 of our species, but there was the greatest incline in massive starlet coral. And by the end of the 6 weeks, our massive starlet coral had the highest rate of algae overgrowth of the 3. We actually haven't found any research to explain why this may be and why the algae overgrowth is greatest in our massive starlet coral, so it's something we are hoping to look into in the future. So what is the take-home message? We found that staghorn coral and mountainous star coral show the best health, healing, and growth in the nursery tree, while massive starlet coral shows the best health, healing, and growth in the wet lab. What's more, we found the most algae overgrowth in our massive starlet microfragments, suggesting that the species isn't ideal for our restoration work here in the Bahamas. Lastly, we found that the reef isn't a suitable environment for growing microfragments of any of the 3 species. This indicates to us that we have to wait for the coral to grow out of its more vulnerable age before outplanting it on the reef. So this is the first time microfragmentation with these 3 species has ever been done in the Bahamas before, so we're incredibly excited to be doing this work. We have the potential to make our coral reefs more resilient to human-caused stressors in the future. Moving forward, we hope that this knowledge will help upscale and improve our restoration efforts in the Bahamas and at CEI. This is our works cited page and the references we use throughout our research. Thank you so much for listening. I just want to say a quick thank you to our research advisors, Valeria and Lily, and our interns, Hannah and Eliza. I don't know if you caught the definition, but we have some coral cookies over here. So they're basically just like dead pieces of coral that we use to like outplant the new corals on. So we're gonna pass this around while we take any questions.
Once you grow the microfragments, how do you plant them back on the reef? And even when they're bigger, when the tides and everything else wash them away?
So they're a little more stable. The question was, wouldn't the current still sweep them away? So once they're bigger, we can get them a little more secure, wedge them in a little more securely, and we can put in a lot of extra effort to fasten them in too, once they're larger.
What will you do with your research?
So as mentioned before, this is a very new process, and like microfragmentation has only been sort of a prominent thing for the past 10 years. So we're just hoping, because we mentioned that the corals, Staghorn coral is actually endangered and the other 2 are major reef builders in the Bahamas. So we're just hoping to build the resilience of the corals. Obviously we can't prevent any of the human-caused stressors. That is something we are hoping to build awareness on. But we are hoping to build the resilience of the coral and find out which species are thriving more and in which, as we mentioned, in which treatment, and to find out which treatment we can grow them at a very young age because of the microfragmentation process. So once they become more resilient in that controlled environment, how we might outplant them on the reef later.
And building off that too, now as we work with our reef restoration, we know the most efficient ways to raise the baby microfragments, so that just makes the process a whole lot quicker.
How old is old? You said beyond the reef, how long do you wait?
So the question was, how long do we wait to outplant them? I mean, as you saw, they already healed their wounds. It did take 6 weeks, but their wounds are 100% healed. So depending on the species, probably like 4 or 5 months for staghorn and a little longer for the other 2, since mountain grows slower.
The nursery location, was that close to the place where you fragmented them from? I mean, the similarity in water changes and that kind of thing, would that have influenced the quickness in which it recovered? It seemed to do better than here in the lab kind of thing. How far away was the nursery from where you harvested?
So the question was, how far away was the nursery from the reef itself? And so it was probably, the nursery trees are around, I would say, maybe like 50 yards away from the reef itself, maybe a little closer, but basically, so they have similar environments, but at the same time, there's very different environments. So like with the nursery tree, we were expecting, and what we found out is there's a lot less predation, because typically the fish that actually eat the corals and the algae on the corals don't venture off from the reef. So therefore, it's kind of like a safe environment for the corals to grow. But considering that they're close together, they do get some similar like current conditions and other things like that. Yeah, go ahead.
Yeah, I was wondering if you measured light intensity between the 3 treatments, and if you made light intensity equal, if you could see similar results.
As of right now, the question was, do we measure light intensity in the 3 treatments? As of right now, we're not measuring light in the wet lab and nursery tree, but we do have a light indicator on the coral reef itself, and we do take data from that.
Yeah, and adding on to that, basically we're trying to figure out the healing and growth at the moment, and we had some other possibilities of factors to change such as water temperature or shading and stuff like that. But because it's so new and it's such a new study that we weren't focusing on adding any other factors to our research study. But it's definitely something that perhaps we could do in the future. Yeah, go ahead.
How would you be able to implement this on a larger scale in the Bahamas? Would you just need more staff to actually perform the process with the microfragments? So I don't know if that's clear or not.
So the question, can you repeat your question? I didn't quite-
So it seemed like you had a really successful growth with what you had, but is there a way to put this on a larger scale, and would that just involve more people doing the process of microfragmentation?
So the question was, how can we do this on a larger scale? So currently, right now, as we said, microfragmentation is really new, and this is the first time we're doing it here. But in the Bahamas, there is something called the Reef Rescue Network, and like I said, the use of coral nursery trees has been used for over 10 years, so there is a network of divers and scientists that do implement nursery trees all over the Bahamas. So this is something that we're working on adding into that.
Listen at your findings of coral reef and Bahamas. That's a local person in Bahama. Somewhat interesting to hear what the damage has been done to the reef. Is the island school looking to inject this information into environmental agency and more or less familiarizing us with the damages at this present time?
So the question was if we're, based on our findings, if we're going to use this information to provide it for environmental agencies locally here in the Bahamas.
So so the question was, will we provide this to environmental agencies? Definitely we'll provide it to reef restoration programs so they can really streamline their efforts. But we didn't really look at the quality of the reefs as a whole or the quality of the reefs here in the Bahamas. So in terms of like advocacy against climate change and anthropogenic factors, it wouldn't really serve much of a purpose. But definitely for conservation efforts, for reef efforts, yeah, it'll be used.